Hey, did you guys know that about 22 billion gazillion years ago, that the aliens invaded Earth and stole a cell and accidentally stole a cow, and to appease the Green Man, the aliens left us cells with blast of chloros that the Green Man would be too busy to breathing oxygen to actually be mad at them? Actually, Charlie, I think you're referring to chloroplast, and they didn't come from aliens. But they do have an interesting history of how they came about. Let me and my friends explain. We were just on our way to give a lecture about this topic to our PLB112 class. You see, Charlie, the chloroplast actually comes from an endosymbiosis between a eukaryotic ancestor and a cyanobacteria, which occurred about 1200 to 2200 million years ago, not a bazillion, gillion, whatever you said years ago. And basically, the eukaryotic cell absorbed the cyanobacteria, and the cyanobacteria became a membrane bound structure within the eukaryotic cell, and it became known as the chloroplast. The chloroplast allowed the eukaryotic cell to perform photosynthesis, and the cyanobacteria could in turn receive protection and nutrients from the cell, being mutually beneficial for both of them. Okay, I get what you're saying. So if I eat a cell, I could start using light and I won't need to eat cat food anymore. Not quite, Charlie, but now that you know a little bit of the history of the chloroplast, let my friend James explain some of the importance and the function of the chloroplast.
Well, Charlie, what's so special about the chloroplast, you may ask? Well, it turns out that chloroplasts are mean green machines, not little green aliens, that work tirelessly to keep the cell alive. The chloroplasts are responsible for initializing photosynthesis through its light reactions, in which the energy coming from sunlight is transferred between the chloroplast's structural components like the chlorophyll, the thylakoids, and the stroma, and eventually converts that energy into the form of ATP and NADH to power the Calvin cycle, or dark reactions that occur to make sugar molecules like sucrose. Chloroplasts have also been found to produce bioactive components like plant hormones or phytohormones, vitamins, lipids, and secondary metabolites in response to environmental stresses that the plant may experience, such as temperature, drought, and pathogens. Even so, chloroplasts can communicate with the nucleus through the process of retrograde signaling to control the expression of genes needed to acclimate to these stressors. So what makes the chloroplast able to do its important jobs? The chloroplast is made up by a stroma, its double membrane, and the thylakoid membrane. The double membrane has remained since before its endosymbiosis with plant cells and is meant to control what comes in and out of the chloroplast. The stroma is a fluid that fills up the chloroplast and houses DNA and the enzymes, like ATP synthase, needed to power the dark reactions that create sugar for the plant. Finally, the thylakoid membrane contains stacks of thylakoids, known as grana, in which within each thylakoid in its lumen is where the chlorophyll pigment is contained to capture sunlight for photosynthesis. Well, Charlie, I hope you got the hang of this now, because Aiden is going to now tell you about how the chloroplast develops in the cell.
What's up, everybody? I'm going to talk about how the chloroplast actually forms inside of a cell. To begin looking at how chloroplasts develop, we have to go back to the all-important resource in plant cells, which is light. Besides the chloroplasts, plant cells have a bunch of different plastids, but we're only really going to focus on the highlighted ones for now. Originally, plant cells house immature proplastids that are like the stem cells of organelles. They don't really have a role yet, but they have the potential to be anything that they want. When grown in the dark, these proplastids transition into etioplasts, which are basically just pale yellow chloroplasts that are photosynthetically inactive and do not contain thylakoid membranes. Basically, the proplastids are infants who can grow up to do anything that they want, and etioplasts are the lazy couch potatoes who need to move out of their parents' basement. Despite etioplast inactivity, once exposed to light, many different light signaling genes are expressed and phytohormones are produced. You might think that this is only important for photomorphogenesis of seeds and larger scale plant growth, but you'd be completely wrong. Light signaling and chloroplast development go hand in hand with each other, since many of the light regulatory genes, such as Hi5, encode for proteins that activate or inhibit chloroplast synthesis. To build off of this, light also affects the levels of many of the hormones that we've discussed in class, including ethylene, gibberellic acid, abscisic acid, auxin, and cytokinin. Auxin and cytokinin are the two that we're going to choose to focus on right now, though. GLK1, GLK2, and GNC are some of the most important genes that activate chloroplast development. They act downstream in both auxin and cytokinin signaling pathways to regulate chloroplast development through formation of the thylakoid membranes. There are hundreds of other genes that are involved in these chloroplast activating pathways as well, but for now we're going to focus on these genes specifically. When you think about cytokinin and auxin, you usually tend to think about how they oppositely function towards root and shoot development, like how we learned with Arabidopsis. However, they actually continue with this opposing relationship into chloroplast formation. It has been established for many years that cytokinin is the major activator of chloroplast development through activation of AHKs, ARRs, and subsequently GLK1, GLK2, and GNC. Cytokinin's pathway to chloroplast development effectively has no inhibitors, which means that cytokinin directly leads to the formation of the chloroplast from etioplasts. Conversely, auxin hates cytokinin with a burning passion and doesn't want to see it succeed at all. That's why auxin tears down what cytokinin is working to build, and what I mean by that is that auxin is involved in the activation of genes such as IAAs and ARFs, which strongly inhibit high-5, GLK1, and GLK2. This prevents chloroplast development when the plant is not ready to maintain photosynthesis. Along with this, since we know that auxin congregates on the shaded side of plant shoots, it is most active when the plant is not receiving light. This leads to the inhibition of chloroplast development in the absence of light. Now I'm going to backtrack to cytokinin for a bit more. If you remember, I said before that etioplasts don't really have much color besides a slight pale yellow. There's one substance in plant cells that is absolutely crucial for the development of the chloroplast from etioplasts. And if you haven't guessed it already, then you should probably switch your clicker answers to chlorophyll. Anyways, cytokinin helps encode chlorophyll biosynthesis genes such as HEMA1, GUN4, GUN5, and CHLM. Increased expression of HEMA1 displays an enhanced level of the encoded glutamyl tRNA reductase, which catalyzes one of the rate-limiting steps of chlorophyll biosynthesis. Basically, cytokinin is responsible for chlorophyll synthesis in plants when subjected to light. However, just like always, auxin is a hater who doesn't want to see cytokinin win. By looking at the color of auxin gene mutants, we can see that they display deep dark green colors which indicate greater presence of chlorophyll. This demonstrates to us that auxin is heavily involved in the negative inhibition of chlorophyll synthesis. Just as a summary, we know that chloroplasts are formed from pale yellow etioplasts which develop from unassigned proplastids in plant cells. Once in their dormant etioplast form, light and phytohormones act to regulate deethylation, which is the transition between etioplasts to chloroplasts. Most importantly, cytokinin acts to promote chlorophyll and chloroplast biosynthesis, while auxin acts to inhibit them. And here you can see in this flowchart all the different pathways that lead to chloroplast development, whether it's abscisic acid, which inhibits chloroplast development, or it's cytokinin, which promotes it. Anyways, thank you for listening, and here are our references.
I see, I see. All the light and all the haters and all the hormones and whatnot. I think we agree we're on the same page here with all these green plants and chloroplasters and whatnot. I don't think we are, Charlie. But you know what? I think you at least learned something here, which has made this all worth it. Now, if you excuse us, we have to go give a lecture on this.